Okay, nice. And we heard about the complicated things. Now it's the pretty part. So I will talk about work that we developed together with Marcelo. Actually, it's a collaboration between us and Texas A&M and NC State. And it's an interactive visualization tool. It's a Shine app to display the results from all the softwares that we are talking about. Let me just put here. Yes, oops. Spoilers. Okay, let me just move the mouse. Yeah, now I have a pointer, and I will keep here this time. Okay. So we were motivated to do this app because we had a lot of phenotypes to evaluate. We had many linkage groups, many population samples, many data sets, and linkage maps and KTLs. And it was really hard to keep all the information in separate scripts. So we thought it would be a good idea to have an app to move through this interactively. And we could also, with that, merge them, and we could connect all our information. And we thought that would be also nice to not need programming proficiency to share this information, because usually we think that in a lab, PhD student or postdoc will run all the analysis, but we always need to share with the PIs or with the entire group, or even share with the community in publications. So, and not everybody needs to know how to do everything, just see the results. And it's also convenient to have a tool to easily get graphics for papers and reports. And also open source one that's not because it's only it's free, this is good, but also because the community could contribute if the code is open. So if other softwares want to be included in VIEWpoly, this is also possible. And another thing that I think maybe is the main thing about this app is to highlight these softwares, these amazing softwares that we are talking about, what they can do. So having a Shine app for that you don't need even to have a data set yet. You can visualize our example and see the capabilities through the graphics. It's a good idea because we can like do some type of advertisement of what our group is developing. So it needs to be easy, so it's easy to install. It's available on CRAN, and it's also available on GitHub. The GitHub now is the updated version. I still didn't submit to CRAN this new version, but I will soon. And once you install either of the versions, you can just open the app with a RAM app. And I also made available a Docker image. So if you don't want to install anything, or if you wanted to easily display in a server, you can use the Docker. And the Docker image is updated. We created tutorials, and there's a very nice and easy tutorial. That's a video tutorial that explains how to do everything that I will tell you here now, and I think the video is even better. So it's still the older version, but there's no much difference in the logic. So you could follow the video, still follow the video. And we have a writing tutorial with print screens and showing how to get everything that you need for making it work. And as I said, it's organized package that we would like contributions. So there's a contribution guideline too. And we also made available this year a branch. So if you want to make as a database, if you want to make your data set permanently available in VIEWpoly and make it available for the public, there's a specific branch that you can do that. And there's a tutorial that explains how to do that too. And this package is organized. So it's a Shine app, it's also a package. And the Shine app is nice because it's an easy way to go from R to a graphical interface. So it's an easy language for doing that. And we also use a Golem framework. That's another package that make this being in models. So it's this package that make VIEWpoly to be very easily expandable. So each one of the tabs that you see on the top, it's a model, and we can expand as much as we want. So it's well organized. And it's a package, so it has a structure of a package, have the description, have examples, and have tests. And okay, like going into the ViewPoly now, the first tab is for imputing the data. And we have an available example there already that you can just explore. That's a potato one that comes with the package. And as I said, we made available a tutorial for making your data sets available permanently. And there's an example here of the image that how would be like it's your data sets instead of the examples, and you just choose one of them. And this is the one that I did for our lab. So me and Jiken, we defined like we didn't know which information we would need in the future, so we put it all of them, like all the phenotypes of the group, the reference genome that was used, which software we used, and the date. So we could track the data set that we are using. So we were like every student that run a KTL or a map, we will have a ViewPoly data set for it and just list this in our computer through ViewPoly. And ViewPoly accepts different type of data. It's in this tab, you will see like a box for linkage map, for KTL analysis, for genome browser, and for Heidecken. That's the new software that was implemented. And for linkage map, we ViewPoly accepts input. Oh, now the mouse is here. Okay, from MapPoly that you just saw, Polymap R. That's also was Peter talk. And I recently implemented also OneMap that opens the possibilities for inbred lines too. And for KTLs, it gets outputs from KTL Poly, KTL R, and Dia KTL. And Heidecken, it's a software that gets results from GWAS Poly, differential expression information, and candidate genes. And the G browser gets all the reference genome information and annotation files. If you click in the box and click in the software that you want to submit your files, there's always some links that connects with the tutorial of the software and the tutorial of ViewPoly on how to get this input and what's this file about. So each one of the tabs has these references. And once you get this file, you just submit it here and submit to ViewPoly. And the same thing for KTL Poly and all the other softwares. And what ViewPoly will do, it will convert all this different type of data sets in ViewPoly data sets. That's a single format. And I'm tracking versions of ViewPoly objects to be able to still like get older versions and convert to new versions, because as you guys saw, these guys made a lot of modifications. So probably the version will need to change, but I want to ViewPoly keep working with old data sets. And so the part of uploading the files, it's a little boring, so but you'd only need to do once, because then you convert to ViewPoly and then you keep only the ViewPoly file. And then you can submit it again in the other tab here. Or you can just keep it in our environment and ViewPoly will identify automatically. And then once you did this is all that you need to do. The rest is ViewPoly will do for you. We'll display all the graphics. So this is an example of the KTL profile that Gabriel just showed you. And the good thing here is that you can just come here and change, come here and change the linkage group, all the phenotypes. So you can select more than one. Select more than one will be like this. And then we'll plot the profile. And we have like some triangles in the bottom. You can select them, and then it will like in the next box will display the effects of the KTLs that you select on the top. You can see these as bars. You can see this as circle, comparing each one of the additive effects. And you can also see the digenic effects that would make more sense for the KTL. So it's different softwares, and ViewPoly identify which one you are you submitted your files, and different graphics can be displayed depending on the features of each one of the softwares. In this case is VIEWpoly. Once you did that, you can select like individuals that have or not the alleles that you are interested according to these other graphics. And in this case, like I'm selecting 2 of them, and I can select individuals that have one allele or can exclude one allele. And it will give me a list of the individuals that have that alleles that you selected in that specific position. And we also have tables for like the breeding values and a summary of the QTL's characteristics. And we always have like the buttons here that you can download the tables. And for the image, I will go back here. You always have like the different formats, and you can just click in downloads for downloading the image, and you can change the dimensions, and you can also change the parent's name in this case. The next table it's about the genome. So it's the connection with the linkage map and the QTL and the genome. So instead of selecting one or more groups, here you only select one, and you have a bar here, showing where the QTL is in this group. And on the bottom of this, you can see the profile just for you to be located where you wanted to explore better. And a relation between the linkage map and the reference genome to see if there is inversions. Because if there is an inversion, you cannot connect with the linkage map with the reference genome. And the next thing is the JBrowser display. So you can see all the features that JBrowser has, but it's in R. So it's the reference genome and the genes. And I'm almost done. So you can see all of this later. The map, it's you can explore one group, and instead of the genes and the genome, it will be the dosage for that region that you selected. And one thing that I would not let behind, it's something that was updated and also proved that VIEWpoly is expandable, because last year I saw a talk here that was Olivia from New Zealand Plant and Fruit Research Institute. And we talked after the meeting, and I said like, oh, and she had this nice Shiny app already. It's like, it will look great in VIEWpoly. And then we worked together in GitHub, just submitting commits and pull requests. And now Heidekan is there. And she talked about this better than me, and she had more time to. And we have the video available. It was in the computational session. Delaney already put in YouTube. So if you want to know more about Heidekan, it's there. Thank you, Delaney. And so the updates, the main updates was some changes, including some features like excluding the haplotypes and defining the parents' names, and I included some small things, but the main things were implementing one map and Heidekan. And the next thing that I'm already started to do, it's implementing power reorigin and multi-population analysis to be able to support what the guys did in VIEWpoly and VIEWpoly. And thank you to all the authors and collaborators and the project members.